thrilled to share that we have Dr. Blank here today with us, and Dr. Blank's topic is gynecologic cancer and antibody-drug conjugates. So antibody-drug conjugates, which are known as ADCs, have been referred to as smart chemo, as it delivered a potent cytotoxic agent to the tumor with minimal damage to normal cells. These new anti-cancer drugs are leading a new era of targeted cancer therapy. Dr. Stephanie Blank, the Director of Gynecologic Oncology for the Mount Sinai Health System, is with us today. We'll discuss how ADCs are holding promising therapeutic potential for gynecologic cancer. In this discussion, she'll also address how ADCs can help fill gaps in the therapeutic landscape, especially for those who are platinum resistant with ovarian cancer. If you do have questions for Dr. Blank, just ask that you write them in the chat, and that way I can ask her throughout the session or towards the end. So Dr. Blank, thank you so much as always for joining us, and the floor is yours.
Thank you. I'm going to share my screen now.
Sure.
Thank you for having me. It's always nice to see some of my friends here. So thank you for being here. I'm going to do my usual kind of spastic speaking. So I'm going to speak today about gynecologic cancer and ADCs, this antibody-drug conjugates. And of course, I'm going to speak very positively about them because I'm very excited about them. And if you're already on them, you may know more than me. I'm speaking very generally. So just my disclosures. Oh my goodness. There's a fire alarm going off in my office here. That's okay. Remember when there was somebody doing my lawn, Jillian, during one of my talks?
I do. We were like, what is that noise? And can everybody mute? But it was you.
Great. Okay. So my institution, I have nothing I can do here.
It's perfectly fine as long if you're comfortable talking over it.
I'll keep talking. I apologize. So my institution does get some, you can't make this up, can you? My institution gets some funding from the companies listed here for trials. I don't get personal funding, or I don't have personal disclosures, and some of what I discuss will be investigational. These are the ABCs of ADCs. I'm going to talk about what they are. I'm going to talk about the ones that are FDA approved for gynecologic cancers, and I'll discuss some of the ones on the horizon, basically. So they all have really long, unpronounceable names, and so I'm going to be using some abbreviations, and I don't usually ever use trade names when I talk about drugs, but I will here a little bit. I apologize. I'm not endorsed by any of these companies, but it's a little bit easier for me because you can see these names are really long. So these are ADCs. Are not brand new drugs. They've been around and FDA approved since 2000, used in other cancers. But the ones that are approved in GYN cancers are, we call this one TV, Tisotumab Vedotin. That's approved in cervical cancer. And Mirvetuximab, I don't even say this, but we call this one MIRV. This is approved in ovarian cancer. And our newest approval, I had to add this on myself to my slide, is Trastuzumab deruxtecan. And this one is approved. It's not approved for one specific tumor. It's approved in all solid tumors that overexpress a biomarker HER2, and I'll speak about that as we go along. So you can see that there's a lot of them, and this for us has really been a class of drugs that we've been able to use since 2021. So what is an antibody-drug conjugate? I have a couple of different pictures, diagrams here to explain. So they basically look like this. There's three parts to them. First of all, they're a monoclonal antibody, and that's the part that looks like a Y. And that is basically a protein that seeks out a specific target on a cancer cell. And then the part that is very chemistry, or I can't explain it all the way because I'm not a chemist, is this linker. It seems really not like a big deal, but it's a very big deal. And that's the part that it attaches the drug to the antibody. And then the drug, and actually the important thing about the linker is it's really stable, in its native form, but once it gets taken into the cell, that's when it breaks down. And then the kind of the big deal of the ADC is this cytotoxic payload that's on the outside here. And that's the thing that really kills the cancer cells once it's taken into the cell. And I don't really like this picture, so I have some other ones. That's like the first one. This one's a little bit simpler. It sort of says the same thing, but again, this is the antibody. This little part is the linker, and then you have your payload. And these payloads are really, really toxic. And when they're sitting here like this, they're like inert. They're not causing any problems. They're not causing any toxicities. But when they go inside, that's when they deliver, and that's when they do things. They have to be internalized. And this thing is highly selective. The antibody. This picture I like simply because it's really the same thing, but the thing that it shows here, which shows the structure and the characteristics. So the antigen, this is the antigen, this is on the cell, on the cancer cell. And the antibody, that's this whole big thing here. That's the point of this. That's the important part. This is the part that makes the drug really targeted. And it is really going to guide the drug to where it needs to go. And then this linker, again, it's really the bridge between the antibody and the drug, and it's going to control the release of the drug. And this site, the thing that's really causing all the problems, these little tiny things, they're almost getting like snuck in there. They're hidden almost. They're getting pulled into the cell, but they're the things that are doing all the work. Any questions about that? I'm going to talk about it a little bit more, but that's sort of like the main thing when you hear antibody drug conjugate. That's the basic structure of all of them. So this is how they work. Basically, the antibody is gonna bind onto this antigen. This is a protein on the surface of a cancer cell. And I'll go through sort of talking what those antigens are. And then once they bind, they're basically taken into the cell. So I kind of made it look like a little Pac-Man here, but they're basically engulfed into the cell. And then once they get inside, the linker releases the payload, the thing that's going to do all the work. So the ADC is degraded and the payload is released into the cell. And then once the payload, is released, it does its thing. It exerts its effects through they're basically two different kinds of payloads. And I think I have a slide in a couple that says this, but either they work either kind of like how platinum works or kind of how Taxol works. They either get in the way of DNA replicating by not letting DNA kind of break down, or by making the tubes of DNA just go straight. And that ultimately leads to cell death. And then the other thing that happens is there's a bystander effect. So even if the drug itself is not taken into a cell, it's almost like ping pong, like this cell that got the ADC hits this cell that didn't, and it kind of still dies. So they kind of, it almost like escalates as it goes. So these things are really potent, because not only is the drug really potent that gets in, but it also ends up affecting cells nearby that did not themselves express the antigen. And so, oh yeah, this is what I was talking about. So the payload, the part that does all the work, is basically two classes of anti-tumor drugs. So they're either microtubule inhibitors, and Taxol is a microtubule inhibitor, or they're DNA damaging agents, and platinums are DNA damaging agents. And these are not Taxol or platinum, but these are like really heavy duty classes of these things that by themselves would be really, really toxic. For example, topotecan is a topoisomerase inhibitor. And when people are on that, their counts get really, really, really low. But when you use it like this, you can get more in, and has less of a systemic effect, basically. And these are some examples of each of those types of drugs, and I'll talk about that in a bit. In general, for these drugs, I'll talk about that in a second. I'm not going to say it right now. So these are some of the parts. The second part of the drug is the part that has basically the ammunition. The first part is the antibody. Oh, here's where I talk about it. So basically, I think of these sort of like tinker toys. So like, do you remember those? I don't know. So one part is the antibody, and one part is you basically stick them together. You stick an antibody, and you stick the linker, and then you stick a payload on the back. So to kind of think about that, these are, I already showed you that there's a lot of currently approved ADCs, but these are basically how they work. Like a lot of them, you can't read this, I'm sorry, but like 1, 2, 3, these are all directed at HER2. A lot of them have like this, and this both have Vedotin on the end. It's basically like pick a antibody and pick a payload and stick them together. So and that's how these generally work. It's not that simple because I don't think they're not all really interchangeable, but it's probably difficult to stick certain ends with certain starts, but this is basically the way that things are working. And the interesting thing about these drugs also is that in some situations, you need to check your tumor cells to see if you express the antigen, and in some you don't. And so I'm going to explain that a little bit as well. So I already told you about the 3 that already exist and are approved for gynecologic cancers. These are all of the ones that are in development. So let's just go 2, 4, 6, 8, 10, 12, 13. That's 13. And there's another one I can think of that's not on here. So there are a lot of drugs in development for gynecologic cancers. And I can tell you that one of the ones that I'm talking about today was approved this year. So the biggest take home that I can give you here today is that there are big advances that are being made that are important. And I think I think those of you who know me know that I'm like optimistic in general, but I do think that the biggest takeaway here is that there are big advances being made. And another thing about these drugs is how you do on drug A, say you received a drug before, doesn't necessarily mean how you're going to do here. So if you don't respond to A, this drug is a totally blank slate. So that's a really good thing. These drugs don't obey platinum sensitive, platinum resistant, so this is all good stuff. I think is very, very exciting. A couple more points about ADCs before I move a little bit on to the real ones. The reason why we theoretically, these drugs, therapeutic window is sort of like how much toxicity you pay for how effective a drug is. You want something to have, you want something to have the lowest toxicity and the biggest effect, but usually you can't have that. Usually more effect, you have more toxicities. So chemotherapy has a small therapeutic window because you have a more effective dose and you have more toxicity. But here you have a wider window because you can basically you don't get so much toxicity with the dose because you can use a lower dose, but it delivers more bang. Okay, now I'm going to move on to the specific drugs that we have that are approved, and then I'll talk a little bit about the other ones. Some of this is a little dry. I apologize for that. Okay, so this is the first one. It's called Tisotumab Vedotin. The trade name that you might have heard is Tivdak. So this is the first one that was approved. This is approved in cervix. And this one is the target is tissue factor, and that is a very widely expressed antigen. So this one you do not have to test for. And interestingly, we also had a trial of this going on in ovary very early on, and it didn't work that well in ovary. Just proves to you that even though tissue factor is very widely expressed, it did much better in cervical cancer than it did in ovarian cancer. And some of these work well in multiple cancers, but this one did not. The other interesting thing here is tissue factor is also very highly expressed in like mucous membranes. It's highly expressed like in your sinuses and in your eyes. And I'll speak momentarily about like that explains some of the side effects that go along with this ADC. So the tissue factor is a protein is very involved in coagulation. Sorry about that. I just kicked my own camera. And it's also very involved in angiogenesis. That's the formation of blood vessels and metastases. And again, it's highly expressed in cervical cancer. So again, this is a monoclonal antibody. It's fully human. It targets tissue factor. There's a linker here, and the payload is MMAE, and this is a microtubule disrupting agent like we spoke about. And I'm not going to go through this. This is just showing, just like the other kind of simpler diagram that I showed you, this gets engulfed in the cell, and it splits apart, and then all these little pink dots are that MMAE that's getting all over, getting over and doing its thing. And so basically there's direct cytotoxicity, there's bystander killing, and there's immunogenic cell death as a result. And I don't know what these two things stand for, so I'm not going to go on with that. Oh, here it is. Antibody-dependent cellular cytotoxicity and antibody-dependent cellular phagocytosis. That means that's the eating, basically the eating of the antibodies. And we talked about tissue factor being in a lot of different places. So the first trial, this is a phase 2 trial. There were phase 1s before. The phase 1 trials are the ones that determine the dose, and the phase 2 trials determine how well something works. And usually, to get an FDA approval, usually you need a phase 3 trial. But this phase 2 trial was so promising that it did lead to an FDA approval, an early expedited, that's the word, expedited approval in 2021 for cervical cancer. And subsequent to this, I hope I'm not like taking away my own thunder, but this was ultimately, so it was first approved based on response rates, and but ultimately it was shown to have improved overall survival, which is a huge deal because these were heavily pretreated cervical cancer patients that previously didn't have a lot of options. This was actually before we routinely used immunotherapy all the time there, and it still works after immunotherapy, but this resulted in improved overall survival, which is very exciting. And these patients had already had Avastin, bevacizumab, but basically these were patients with recurrent or metastatic cervical cancer, and they received this drug. And then what we see here, this is called a waterfall plot. When it goes down, the tumor is shrinking, so you can basically see that the majority of people had their tumors shrinking. You have to go down more than 30% to have it be considered a response. And so here, this is a 25% response, but that's actually very good here in this situation. And so that was FDA approved, as I said. When you talk about the toxicities here, it does cause hair loss. 30% of people had significant nosebleeds, as I mentioned before. It's big in the sinuses, and conjunctivitis and dry eye were very big. So these were actually pretty significant side effects, just different from what you would think about when you think about chemotherapy. But anyway, this is recurrent cervical cancer is a hard cancer to treat. So this was a big revelation, and really the first ADC approved for gynecologic cancer. And now we're going to move on to the first one approved for ovarian cancer. So this was really, this is like confetti, because this was a huge deal. This was in November 2022 when the FDA granted accelerated approval to Mirvetuximab for folate receptor alpha positive, and I'll talk about that, platinum-resistant epithelial ovarian, fallopian tube, or primary peritoneal cancer. This was again a very big deal, and I'll sort of go through it a little bit more. So what MIRV is, and the trade name here is Elahere. This is similar to the very initial picture I showed you. So the antibody is to folate receptor. So here in this one, you do have to be tested. You do have to get your tumor tested for folate receptor. You have a linker, and then your payload is called DM4, and again, it's another tubulin inhibitor. And so I think pretty soon I'll explain what the folate receptor is, but it is expressed on 90% of ovarian cancer cells, but it's important, it has to be highly expressed. And so MIRV is really similarly to all the other ones, it comes into the cell. If there's a folate receptor there, it's internalized, then it's degraded, and then it disrupts the tubulin, and then the cell dies because it stops being able to reproduce. So folate itself is a B vitamin. It's used in metabolism, and it requires active transport to get into cells. Folate receptor alpha is the receptor that assists taking the folate into the cells, and it's limited on normal cells, but upregulated in cancer. And then the DM4 is highly potent, and only a very small effect, a small amount drives the effect. And they use a lot of war-related. You probably, if you ever see a talk on ADCs in general, there's all these war-related Trojan horse. That's what people talk about it like. So the study that really led to the FDA
Dr. Blank,
approval
I can't see your slides anymore. I'm not sure if it's just me for some reason, but I can't.
Nobody see it?
Oh, you know what? No, I apologize. I hit something on my screen. I apologize.
Okay, sorry. The study that led to the approval here was called MIRASOL, and it basically included patients that had platinum-resistant disease, and they had folate receptors there, and it had to be 2+, and I'll talk about that in a second. They had been, they could have pretty platinum-resistant disease, a lot of prior treatment. They could have prior Bev and PARP, and they were randomized to either MIRV or chemo. And again, the waterfall plots, you can see this looked, better than investigator choice chemo. 42% overall response rate, which is very good for platinum resistant disease. This is progression-free survival. And if you want to just say this in English, they had what do we have here? Hazard ratio 35% less chance of progressing. And this is overall survival. They had a 33% increased chance of living. And when this was presented, it was presented as a new standard of care. And it was the first treatment to demonstrate a benefit in overall survival in platinum-resistant ovarian cancer. So lots of times people might go a longer time with their cancer staying away, or they might have a better chance of their cancer shrinking, but it might not, it hadn't in the past necessarily led to people living longer, but this one did. So this is a really big deal for people with recurrent platinum-resistant ovarian cancer. So a statistically significant and clinically meaningful improvement in progression-free survival, response rate, and overall survival compared to chemotherapy, a differentiated safety profile consisting predominantly of low-grade I'm going to speak about that in a minute, and gastrointestinal events. And it was the first ADC for ovarian cancer. And a new standard of care. This is very exciting, and I'm going to speak a little bit more about it momentarily. I just bringing this up. We have a trial here looking at it in a maintenance setting after platinum-sensitive recurrence. So we're still looking at where this goes, but I really like it as treatment for platinum-sensitive, but platinum-resistant cancer with the folate receptor alpha. All right, so let's talk for a minute about folate receptor alpha testing. You can have the testing done at any point, and at some point it was free. It changed, like Caris used to be free, and now they're charging. But there are some different places that are charging for it. But it's something that you can ask. Just that your initial tumor can be tested for it. And it can be old tissue, and you could just ask that it be done. It could just sit there. Even if you don't need it right now, you can have it be done. So again, 90% of people, this one says 76 to 89, but about 90% of ovarian cancer is going to have overexpression of folate receptor alpha, and it's expressed in some non-malignant tissue. High expression is in 35%. So this is really important because for Mirvetuximab, you have to have high expression. There was an earlier study looking at Mirvetuximab. Sorry, this got messed up here. And they used medium and high as entry criteria, and that study was negative. So it's very important that the correct people are chosen to get this drug, that's how it will work. So you really need to have this 2+ or higher expression. This is what it looks like, low, medium, and high when they stain it. So that's what has to happen for you to be able to get this drug. If you take this drug and you don't have this overexpression, it's not going to work. And this is what the report looks like if you get it tested and you're positive. They'll show you there. This one's negative. Sorry, negative. This, I don't know why I have this here. This is positive. So folate receptor can have some off-target effects, and the biggest one, and this is scary for people sometimes. So if you have biggest side effect is ocular toxicities, eye problems. You see your eye doctor very frequently while you're on this medication. You can't wear contact lenses. That would be difficult for me. And you need to be on eye drops a lot. They tend to be reversible. So if you go on this and you start developing that, first of all, you can drop the dose. You hold it, you drop the dose. It still works just as well if you do those things. But this is a side effect that people get a bit concerned about. The GI things, there can be some nausea, but it's not bad, and there's not as much hair loss as there is with the one that I just mentioned prior. So this one has been pretty exciting for people in general. And now I will move on to our newest member of our trio here. And that one is Trastuzumab deruxtecan, also known as Enhertu. And this one is based on HER2 testing. An important thing about HER2 testing is that most GYN patients might not have had HER2 testing done on their tumor. So similar, this is another one where you need to have the biomarker tested. You need to have your tissue tested for it. The only people that generally have HER2 testing in GYN cancers are people with serous endometrial cancer, because we would use a specific drug for that. So this is something that you can ask to have tested. There's also, if you have your tumor sequenced, there's a correlative gene that you could have a mutation in that also would make you eligible for this drug. So then, and the third thing about the biomarker is that you can see here, it's gastric guidelines that are used to determine positivity, not breast guidelines. So it's a little more lenient to get a 2+, to get positivity here. So 2+ can be considered negative for breast guidelines. So it's very important that the scoring is looked at very carefully to determine positivity. Something to discuss with your doctor. Anyway, this is another one you can just say, please have my tumor tested for HER2, just so you know. Anyway, so this ADC was tested in a whole bunch of different, in all these kinds of tumors. It was this trial was called the Destiny Pan tumor test, and this is really this is unusual. This was basically they only had a couple of they only had 40 patients in each of these cohorts. Cervical, endometrial, and ovary. These are the ones I'm interested in. You'll probably see some data on these that I'll show you, but I'm only interested in these. And I ovary circled, but I'm going to talk about all of these. So basically, this drug uses HER2, and then they use a topoisomerase inhibitor payload. So that one again is a tubulin thing, and there it is. And we gave it here when this is the testing that people had in general for the HER2. Oh, there's the picture of it. So one thing I haven't talked about is when you have these antibody-drug conjugates, they report these things called DAR, which is the drug-antibody ratio, and the higher the number means it's delivering much more of the drug. So this is a pretty high drug-antibody-drug ratio, which means it's delivering a lot more bang for the buck, basically. So cervical, endometrial, ovarian. So this is basically overall rate of response with 3+ and with 2+. So remember, this is only 40 patients, so it might, for each of these cohorts, it might not be so much, but our tumors, our gynecologic tumors responded so well here. We're 85% response rate, 75% response rate. These are really unheard of. And so this is why we're testing everyone for HER2 now, because if people are positive, we're really giving people these drugs. And I have to say, people have been doing really, really well on them. So this is just very exciting. And we don't usually, we don't even usually get approvals based on response rate, as I said before when I was talking about the TV. So this has been extremely exciting. And when the other thing is, this is that data again, and this is the changes in the size of the tumor, just really marked. Look, I mean, the waterfall plots are so far down. Here, we previously thought that the rates of expression of HER2 in these tumors was much lower than it actually ends up being. So previously these numbers were thought to be significantly lower. So like this is really pretty impressive. So it's definitely worth getting tested, and it can be even in very heavily pretreated people. And as I mentioned, some people with endometrial cancer, serous cancers, who are HER2 positive might have previously had trastuzumab, which is also called Herceptin, but this still works there. So this is a very exciting advancement. And on April 5th this year, the FDA granted accelerated approval to this drug for people with unresectable or metastatic HER2 positive solid tumors who had had prior systemic treatment. So it's second line. It's not first line, but it's been very, very good. So this one, we're super excited about these drugs in general. You might hear it in my voice. So I'm going to speak now just about a couple of other new ones. It's going to be, it might not mean so much because to me, I'm going to be saying names I can't really say, and I'm going to show you antibodies. I'm going to show you their payloads. Not going to mean all that much, but just look at the waterfall plots and just say to yourself, oh, there's a lot of them. Okay. So I'm going to show you, I think, two that are also against folate receptor alpha. Okay. And the other thing is you can't necessarily say, well, if you were on Mirvetuximab and it stopped working, that it's not going to work for these, because it can still work. So it's a different mechanism of action. So this one, I guess it's called Luvelta. This is another one with MIRV. It has, that's to folate receptor alpha. It has a different payload. It has a DAR. Remember, we just talked about that, of 4, and it has, this nice waterfall plot. And this one is currently moving to a late phase trial, which means that it's done well, like here. So this one is actually pretty far along in moving towards approval. This one, farletuzumab, I actually can say that one is another folate receptor alpha 1, and this one too, a lot of good responses. And this is another microtubule inhibitor, another DAR of 4. This one, I can't remember what we call this one, Ralu, I think. I don't remember what we call this one. This one is super exciting. This one, CDH6, you do not have to have testing for. This is a highly expressed, I don't know, antigen. And this is being looked at now, I believe, in the front line for ovarian cancer. And this one also, it has a very high DAR, but this one's been very, very positive. I saw this presented when it was presented, and people were extremely excited about it. It was presented in a phase 1 setting at SGO either this year or last year. And then finally, I'll just talk about these two. These are TROP2 ADCs, and TROP2 is highly expressed in a lot of cancers. You don't need to be tested for these. And this is a picture from ASCO that I took. So these are both SASI is the first, Sacituzumab is the first part of both of them. This one is Sacituzumab, I can't say it, Vedotin, and this one is Sacituzumab, whatever that is with the T. But these are both very exciting, and we are actually going to have, we have a trial with just opening, this one in platinum-sensitive ovarian cancer and this one in endometrial cancer. So these are all just very exciting drugs, and some of them you need to have a certain biomarker, some of them you don't. Again, and I think that there's obviously a lot of them coming around. Now, how are they going to advance in the future? We need to understand if some, I've had some patients where they work great and then suddenly they don't, and why is that? Maybe the antigens are regulated, or maybe the binding site doesn't work as well. Maybe they're not like scooped in quite as well. Maybe they're pushed out, and maybe something's happening with DNA repair. We don't know, so we have to learn a little bit more about that. Just again, they're a growing part of our armamentarium. They're from one, on and on and on. I think this era is going to continue to go up. These are ours. And, if you've ever heard me speak on one of these things before, I know I sound like a broken record, but clinical trials is how this all happened. Clinical trials, we were involved in clinical trials for all of these, we weren't involved in clinical trials for the HER2 one, because they were so small. We involved it for the first two. And, we've really seen things change. We know that clinical trial participation is associated with improved overall survival. This is just one demonstration of that in patients with platinum-resistant ovarian cancer. We know that there are benefits to clinical trials. You access new drugs. You have very close monitoring of your health. You have an active role in your healthcare, et cetera. It's an opportunity to make a valuable contribution to cancer research. Can't speak more about clinical trials. Any of you on here that have been in clinical trials, thank you, and thank you all for your attention. Appreciate it.